Analyzing the impact of non-coding variants with OpenCRAVAT. OpenCRAVAT performs variant interpretation that includes evaluation of variant impact, functional annotations, and scoring methods. Since the majority of human genetic variants occur within non-coding regions of the genome, the App Store includes dozens of resources that could help identify disease-relevant non-coding variants. Types of non-coding annotators. There are 4 types of non-coding annotators: functional elements, clinical relevance, evolutionary constraint, and sequence properties. These are the annotators related to non-coding functional elements. In contrast to coding variants, which impact genes and directly influence the amino acid sequence of a protein, non-coding variants may impact one of several types of non-coding functional elements. While the entire catalog of functionally relevant non-coding DNA has not been fully characterized, OpenCRAVAT includes annotation sources that can be used to identify variants which occur within known functional elements in the non-coding regions of the genome. OpenCRAVAT currently includes annotators that specify the locations of promoters, enhancers, non-coding RNAs, and transcriptional binding sites. In addition to these catalogs of known functional elements, the Segue annotator is an exceptional source of predicted human regulatory elements. Segue uses a conservation-associated activity score to create an encyclopedia of human transcriptional and regulatory elements. This Segue annotator group includes both the cell-type agnostic Segue annotations and 25 additional cell-type specific predictions. In the main Segue annotator, the scores provide a measure of importance, where higher scores are associated with increased functional activity. For particular cell types, Segue provides the annotation label for each variant that describes what kind of functional or non-functional region the variant is predicted to occur within. In combination, these annotation sources can help identify the direct functional impact of non-coding variants. The second type of non-coding annotator is clinical relevance. These are the annotators related to clinical relevance, including clinically relevant variants, EQTLs, and databases of GWAS data. Databases of clinically relevant variants include both coding and non-coding variants that are directly related to the disease. GWAS and EQTL loci are more frequently non-coding but do not directly cause disease. Collectively, these annotation resources can identify variants that are known to be related to disease. In general, these variants are more well described in the literature compared to the average non-coding variant. These are the annotators related to evolutionary constraint. Variants that occur at positions of the genome that have undergone fewer changes between species are considered more conserved evolutionarily. High conservation of a nucleotide indicates that the sequence has been maintained by natural selection, and that the mutation of that nucleotide may negatively impact fitness. These methods provide scores that reflect the amount of nucleotide diversity in humans compared to closely related organisms. Each tool varies in their method of calculating an evolutionary score. These tools can give insights to the non-coding variants that are shared among multiple species, and can find variants that impact nucleotides that are unlikely to be impacted by chance. These are the annotators related to sequence properties. These properties include the identification of pseudogene and repeat sequences, and annotations of haplotype blocks. Variants within pseudogenes or insertion-deletion variants in repeat regions may be less likely to be clinically relevant, or variants within the same haplotype block tend to be inherited together. Haplotype blocks are highly variable depending upon ancestry, so the haploreg annotator includes haplotype blocks for different populations from the 1000 Genomes Project. These properties do not directly translate to functional or clinical importance for a variant. Instead, these annotations provide insights to the context of a variant by describing the type of DNA sequence being impacted. Using non-coding annotators in OpenCRAVAT. Next, let's look at an example of how these annotations can categorize non-coding variants. This example file, example.vcf, is available in the description below and includes 867 variants from chromosome 1. Open the web server at run.opencravat.org and run a job using the provided VCF file. Select genome version HG19 and click the checkboxes for encode TFBS and Linsight. After the job is complete, open the results viewer by clicking the button in the Jobs table. On the Summary tab, we can learn about the variants in this file. The coding/non-coding summary widget shows that more than half of these variants are non-coding. The sequence ontology summary widget shows that many of these variants are intergenic, and 256 variants impact link RNAs. On the Variant tab, the Sequence Ontology column can identify variants that impact non-coding RNA sequences cataloged in the Ensemble database. There are several dozens of variants that impact non-coding RNAs. In fact, this variant impacts an mRNA and is annotated by encode TFBS to be a binding site for the transcription factor called CTCF. However, the line site score is 0.048. The line site score corresponds to the probability of negative selection, and this value is low. This means that the nucleotide has not been subject to negative selection and is more likely to have substitutions in the human lineage. So, this variant impacts a nucleotide that is less likely to be an important and conserved part of that transcription factor binding domain. Variants that impact functional regions of the non-coding genome may be more likely to be disease relevant. Supporting information such as evolutionary conservation, allele frequency, or sequence features can inform on the likelihood of that variant to directly or indirectly influence disease. The App Store is updated with new annotation sources frequently. To get started annotating and analyzing non-coding variants with OpenCRAVAT, go to run.opencravat.org.